Hello, my lovely anatomists and physiologists. Welcome back. We are talking about glomerular filtration rate, or what we can abbreviate as GFR. And this is talking about the volume of filtrate formed by both kidneys per minute. And we're usually seeing about 80 to 140, this is depending, of course, on body type, per 80 to 140 milliliters per minute. This is filtrate, not urine. So this is actual getting that material out of the blood and into that capsular space. Or it might make more sense for you to say 150 to 180 liters per day, with the knowledge that you're only producing 1 to 2 liters of urine per day. So clearly, we're pushing out way more fluid from the glomerulus than what we are actually making to exit the body, right? So there's going to be a lot of like returning of material, or what we're going to be talking about as reabsorption, and we'll define that again in the future. Okay, now when we're looking at our model here, we're seeing, of course, our glomerulus, which is our fenestrated capillary. Let me make my font smaller so it's easier to read. And we're looking up here at Bowman's capsule, or you can call it the glomerular capsule. And remember, we have this space called the capsular space. And so we said that the job of the capsule is filtration. And this is where pressure is going to push fluid and solute out based on size. So that's my real abbreviated description of filtration that we talked about before. And so what we do need to look at is different pressures and how those pressures are determining or playing a role here in the glomerular filtration rate. So we're going to be looking really at three. So we have what's called the blood hydrostatic pressure. We have what's called the blood colloid osmotic pressure. And then we have the capsular hydrostatic pressure. And let's also mention here the capsular colloid osmotic pressure. So let's talk about these three. So let's first tell ourselves, anytime we see hydrostatic, for us here, just think about like blood pressure. Just think about force from the heart, force generated by heart. And then anytime you read the word colloid, remember this is a suspension of proteins. And then whenever we talk about osmosis, that's the movement of water from a concentrated to dilute solution. I said it backwards. Dilute to concentrated. Okay. So hydrostatic is force generated by the heart. That's like our blood pressure. I think we already have a concept there in our minds that works really well. Colloid is talking about suspension of proteins, right? And then osmosis is the movement of water from a dilute solution to a concentrated solution. So when we talk about the blood hydrostatic pressure, this is literally the blood pressure inside glomerulus. The glomerulus, remember, is the fenestrated capillary bed that's inside the capsule. And we're going to see that the glomerular pressure, the blood hydrostatic pressure here, which instead of blood hydrostatic pressure, you might see it listed as glomerular. So when we're looking at the blood hydrostatic pressure, the glomerular hydrostatic pressure, we're going to see it's higher than systemic blood pressure. And part of this is because the afferent arteriole is going to have a larger diameter than the efferent arteriole. So the afferent arteriole is larger than the efferent arteriole. So if you have like a big opening and then the exit is smaller, you're building up pressure in the system, you're backing it up. And then we also have the role of the mesangial cell, which can help with this constriction as needed. So we're going to see what we can call the blood hydrostatic pressure really being the force for filtration. So we can say blood hydrostatic pressure, or instead of blood, we can say glomerular hydrostatic pressure is really going to be the force to push material out. When we talk about the blood colloid osmotic pressure, what we're going to see here is that you're going to have plasma proteins such as albumin becomes concentrated. So you're going to see red blood cells do not leave the fenestrated capillary, so they don't leave the glomerulus. White blood cells are not leaving here. Platelets are not leaving here. And plasma proteins such as albumin are too big to leave here. So if you get initially this mass exit of fluid, electrolytes, and so forth out into that capsular space because of the force from the heart, because of the high blood hydrostatic pressure, you'll start to get your blood becoming sort of concentrated. And so you can imagine that this is going to pull water back in. So it's not going to be as high of a force as the blood colloid osmotic. Excuse me, I'm saying all the wrong words. It's not going to be as much of a driving force as the blood hydrostatic pressure, but we'll see a little bit of pull of fluid back into the glomerulus based on the blood colloid osmotic pressure. So we have two forces, they're opposing each other. Blood hydrostatic pressure is pushing fluid, electrolytes, nutrients out. Anything that can fit through the fenestra and the filtration slit is leaving. As that happens, the stuff that remains becomes concentrated, and so that's going to pull some of that water back in. Okay, then we have what is called the capsular hydrostatic pressure. So what we'll see is there is going to be fluid. The capsular space is not empty. Some fluid is present. In the capsular space. I suppose if you're dehydrated, then this space might be empty, right? Depending on level of dehydration. But like a normal, healthy, functioning, eating and drinking adult, I imagine there's going to be a little bit of fluid that's here all the time. And so because this is not an empty space, there's going to be a little bit of resistance. So this creates, let's say, some resistance to filtration. Because some fluid is already present, not all the fluid can exit, right? So let's go back to our drawing. And let's write in the capsule, the capsular hydrostatic pressure, and draw the arrow like kind of pushing into the bloodstream. So we've got these three forces and they're different. So the blood hydrostatic pressure is forcing fluid and solute out into the capsular space. So I have my arrow from the glomerulus out to the capsular space, and it's my biggest arrow. Then I have the blood colloid osmotic pressure showing the arrow back into the glomerulus. So as fluid and electrolytes leave the glomerulus, what gets left behind becomes concentrated, and that's going to pull fluid a little bit back in. And then also I have a third pressure, the capsular hydrostatic pressure. And here I'm showing the arrow pointing back into the glomerulus. The idea here is the space is not empty. So there's going to be some resistance to like this mass exit. I wrote about this fourth pressure here. So I also have listed what's called the capsular colloid osmotic pressure. And this would be talking about proteins in the capsular space. Those proteins should not be present in the capsular space. You're going to get amino acids, which are the building blocks for proteins, but not large proteins themselves. If you do have proteins present in this space, you've got damage to the kidney. And so this should be at 0. So it shouldn't actually be involved in the whole overreaching kind of conversation. What we're trying to figure out here is what is called the net filtration pressure. And so that's what's actually going to exit. We're going to call it net filtration pressure. And we'll see it's going to be about 10 millimeters per mercury. And so it's combining. So you have this really high number for the blood hydrostatic pressure, but then you subtract the blood colloid osmotic pressure and you subtract the capsular hydrostatic pressure. And so the net filtration pressure is just around 10. So it's not super high like it would be if you didn't have these other forces that were counteracting. Okay. So that is actually it for our discussion of the glomerulus and the glomerular capsule or Bowman's capsule. And what we're ready to do next is take a look at that tube called the proximal convoluted tubule. So stay tuned, and as always, take care of yourselves and each other.